this video, we are going to learn about the human perspective, that is, the role of cell adhesion in inflammation and metastasis. Inflammation is one of the primary response to infection, which we all know. If a part of the body becomes contaminated by bacteria, as might occur following a puncture wound to the skin, the site of injury becomes a magnet for a variety of white blood cells. White blood cells or leukocytes that would normally remain in the bloodstream are stimulated to transverse the endothelial layer that lines the smallest veins or venules in the region and enter the tissue. Once in the tissue, the leukocytes move in response to chemical signals toward the invading microorganisms which they ingest. Although inflammation is a protective response, it also produces adverse side effects such as fever, swelling due to fluid accumulation, redness, and pain. Inflammation can also be triggered inappropriately. For example, damage to the tissues of the heart or brain can occur when blood flow to these organs is blocked during a heart attack or stroke. When blood flow to the organ is restored, circulating leukocytes may attack the damaged tissue, causing a condition known as reperfusion damage. An overzealous inflammatory response can also lead to asthma, toxic shock syndrome, and the respiratory distress syndrome. A great deal of research has focused on questions related to these conditions. How are leukocytes recruited to sites of inflammation? How are they able to stop flowing through the bloodstream and adhere to vessel walls? How do they penetrate the walls of the vessel? How can some of the negative side effects of inflammation be blocked without interfering with the beneficial aspects of the response? Answer to questions about inflammation has focused on three types of cell adhesion molecules: selectin, integrins, and IGSF, that is immunoglobulin superfamily proteins. A chain of events that has been proposed to occur during acute inflammation is shown in the figure, where when endothelial activation is happening, then neutrophil trapping is happening, neutrophil is activated, neutrophil adhesion is happening, neutrophil invasion is happening. Okay, which is done by ICAM, selectin, platelet activating factor, integrin. Integrin is something which is inactivated, and then the integrin activated type. These are the steps in the movement of neutrophils from the bloodstream during inflammation. A chain of events that has been proposed to occur during acute inflammation can be seen. The first step begins as the walls of the venules become activated in response to chemical signals from nearby damaged tissue. The endothelial cells that line these venules become more adhesive to circulating neutrophils, a type of phagocytic leukocyte that carries out a rapid, non-specific attack on invading pathogens. This change in adhesion is mediated by a temporary display of P and E selectin on the surface of the activated endothelial cells in the damaged area. When neutrophils encounter the selectins, they form transient adhesions that dramatically slow their movement through the vessel. At this stage, the neutrophils can be seen to roll slowly along the walls of the vessel. A number of biotechnology companies are attempting to develop anti-inflammatory drugs that act on interfering attempting to develop anti-inflammatory drugs that act by interfering with the bonds of ligands to E and P selectin. Anti-selectin antibodies block neutrophil rolling on selectin-coated surface in vitro and suppress inflammation and reperfusion damage in animals. A similar type of blocking effect has been attained using synthetic carbohydrates like epomycins that bind to E and P selectin, thereby competing with carbohydrate ligands on the surface of the neutrophils. As the neutrophils interact with the inflamed venule endothelium, interactions take place between other molecules on the surface of the two types of cells. One of the molecules displayed on the surface of the endothelial cells is a phospholipid called platelet activating factor. Platelet activating factor binds to a receptor on the surface of the neutrophil, sending a signal into the neutrophil that leads to an increase in the binding activity of certain integrins like alpha L beta 2 and alpha 4 beta 1 already situated on the neutrophil surface. This is an example of a type of inside out signal that is illustrated. The activated integrins can bind with the high affinity to IGSF molecules ICAM1 and VCAM1 on the surface of the endothelial cells, causing the neutrophils to stop their rolling and adhere firmly to the wall of the vessel. The bound neutrophils then change their shape and squeeze between adjacent endothelial cells into the damaged tissue. Invading neutrophils appear capable of disassembling the adherence junctions that form the major barrier between cells of the vessel wall. This cascade of events, which involves several different types of cell adhesion molecules, ensure the attachment of blood cells to the walls of blood vessels, and subsequent penetration will occur only at sites where leukocyte inversion is required. The importance of integrins in the inflammatory response is demonstrated by a rare disease called leukocyte adhesion deficiency, LAD. Leukocyte adhesion deficiency. Persons with the type 1 form of leukocyte adhesion deficiency are unable to produce the beta 2 subunit as a part of a number of leukocyte integrins. As a result, the leukocytes of these individuals lack the ability to adhere to the endothelial cells of venules, a step required for their exit from the bloodstream. These patients suffer from repeated life-threatening bacterial infections. The disease is best treated by bone marrow transplantation, which provide the patient with stem cells capable of forming normal leukocytes. Administration of antibodies against the beta 2 subunit can mimic the effects of LAD, blocking the movement of neutrophils and other leukocytes out of blood vessel. Such antibodies might prove useful in preventing inflammatory response associated with diseases such as asthma and rheumatoid arthritis or with reperfusion. Cancer is a disease in which cells escape from the body's normal growth control mechanisms and proliferate in an unregulated manner, were the malignant cells to remain in a single mass, as often occur in some type of skin cancer or thyroid cancer. Most cancers would readily cured by surgical removal of the diseased tissue. Most malignant tumors, however, spawn cells that are capable of leaving the primary tumor mass and entering the bloodstream of lymphatic channels, thereby initiating the growth of secondary tumors in other parts of the body. The spread of a tumor within the body is called metastasis and is the reason cancer is much a devastating disease. Metastatic cells, cancer cells that are able to initiate the formation of secondary tumor, are thought to have special cell surface properties that are not shared by most other cells in the tumor. For example, metastatic cells must be less adhesive than other cells to break free of the tumor mass. So we can see a cancer cell is there and the BM is there and cancer cells are migrating. So muscular basal membrane and cancer cell will be migrating from there. Steps leading to the metastatic spread of the epithelial cancer. A carcinoma. A fraction of the cells of the primary tumor lose their adhesiveness to other tumor cells and gain the capability to penetrate the basement membrane barrier that underlies the epithelial tissue. These cells, which have assumed a mesenchymal-like appearance, migrate through the surrounding stromal tissue and cross the BM of a blood or lymph vessel, thereby entering the general circulation. The cells are carried to other tissues where they migrate back across the BM of the vessel and enter a tissue in which they possess the potential to form secondary tumors. Only a very small percentage of tumor cells that are released from a primary tumor manage to overcome these numerous hurdles, but those that do pose a threat to the life of the host. The circulating tumor cells have been isolated from a blood sample of a patient with prostate cancer, even though the blood of a cancer patient may contain less than one cancer cell for every billion normal cells. These rare cancer cells can be selectively trapped on a chip that has been coated with antibody molecules directed against a cell surface protein, in this case EPCAM, that is present in the cancer cells and absent from normal blood cells. Metastatic cells must be able to penetrate numerous barriers, such as the extracellular matrices of surrounding connective tissue and the basement membranes that underlie the epithelium and line the blood vessels that carry them to distant sites. Although the presence of cancer cells in the bloodstream is potentially lethal circumstance, and it does provide an opportunity for non-surgical access to a patient's cancer. A number of devices has recently been developed to trap rare circulating tumor cells present in blood sample taken from cancer patients. Once they have been captured, CTCs from a patient can be studied to determine the molecular characteristics of the tumor, how aggressive the tumor is likely to be, how it might be best treated, and how well a particular therapy is progressing as reflected in the diseased number of CTCs over time. One day, CTCs might be used as the basis for early detection of disease. Metastatic cells must be able to invade normal tissues and survive to form secondary colonies. For some reason, this third step appears to pose the greatest obstacle in the process of metastasis for most types of tumors. Consequently, only a very small fraction of cancer cells that exist in the bloodstream typically produce viable secondary tumors. The mechanisms used by cancer cells to penetrate extracellular matrices is poorly understood because such events have been virtually impossible to study within the tissues of a living animal. It is thought that movement through the basement membrane is accomplished largely by ECM digesting enzymes, most notably the matrix metalloproteinases. These enzymes are presumed to degrade the proteins and proteoglycans that stand on the way of cancer cell migration. In some types of cancer cells, MMPs are directly associated with long cell projections called invadopodia that push their way through the ECM. Among other tumor promoting functions of MMPs, cleaving of certain portions of the proteins of the ECM or cell surface produces active protein fragments that act back on the cancer cells to stimulate their growth and invasive character. Because of their apparent roles in the development of malignant tumors, MMPs become a prominent target of the pharmaceutical industry. Once it was demonstrated that synthetic MMPs inhibitors were able to reduce metastasis in mice, a number of clinical trials of these drugs were conducted on patients with a variety of advanced inoperable cancers. Unfortunately, these inhibitors have shown little promise in stopping late stage tumor progression and, in some cases, have led to joint damage. To date, the only FDA approved MMP inhibitor, periostat, is used to treat periodontal disease. Changes in the number and type of various cell adhesion molecules and the stability of cells to adhere to other sites, cells, or to extracellular matrix have also been implicated in promoting metastasis. The greatest focus in this area has centered on E-cadherin, which is the predominant cell adhesion molecule of the adherent junctions that hold epithelial cells in a cohesive sheet. Loss of E-cadherin from epithelial cells during certain events of embryonic development is associated with conversion of the cells into a less adhesive, more motile mesenchymal phenotype. A remarkably similar epithelial mesenchymal transition occurs at the peripheral edge of a tumor as malignant cells separate from the primary tumor mass and invade the adjacent normal tissue. This is an important step in the process of metastasis. Surveys of a, surveys of a variety of epithelial cell tumors, breast, prostate, and colon cancers confirm that these malignant cells have greatly reduced levels of E catherine. The lower the level of expression of E catherine, the greater the cell's metastatic potential. So, when E catherines are lower, the metastatic potential, the ability of tumor cells to migrate will be increased. Conversely, when malignant cells are forced to express extra copies of the cadherin gene, the cells become much less capable of causing tumors and then injected into host animals. The presence of E. cadherin is thought to favor the adhesion of cells to one another and suppress the dispersal of tumor cells to distant sites. E. cadherin may also inhibit the signaling pathways within the cell that lead to tissue invasion and metastasis. The importance of E. cadherins is evident from a study of family of native New Zealanders that had lost 25 members to stomach cancer over a 30-year period. Analysis of the DNA of the family members has revealed that susceptible individuals carry mutations in the gene encoding E. catherine.